Hi guys, Kenna here. So today we're going to do kind of an overview of the whole protein synthesis process. So we've done transcription by itself, translation by itself. Now we're really going to try and put all the pieces together to give you the big picture view of the process of protein synthesis. That being said, the essential questions for today's lecture really are a combination of the ones from the previous lectures with just one more added together. So let's go over those essential questions now and then go ahead and do our quick overview. Number one, where does transcription take place? At this point, you should probably be able to answer all of these except maybe the last one. Number two, what are the similarities and differences between introns and exons? You'll remember these from our discussion of transcription. Number three, where does translation take place? Again, you should be able to answer this definitely. Number four, what is the role of all three types of RNA? We've talked about three now, but we've really emphasized two more than the third type. Number five, what must happen following the production of the polypeptide in order for us to get to the end of protein synthesis? And number six, how do the steps of the process determine the final protein produced? We've spent a lot of time this semester talking about the relationship between structure and function. And so this is no more or less true than it is here when we're looking at protein synthesis. Remember that proteins do so many different jobs around the body that we need to remember that they fold into these different structures, both tertiary and quaternary, that give them the ability to perform this wide array of tasks. Okay? Everything from motion, when we start thinking about myosin actin, all the way up through our hormones, through our enzymes and catalysts, even to structural pieces when we start looking at things like keratin and collagen. So proteins do a lot, and that is largely because of the ability to form all these different structures due to the folding process that actually occurs after we go ahead and make the amino acid sequence of the polypeptide. So let's review what the three main types of RNA are, and then we'll talk about how they're used throughout this process of protein synthesis. There are three main types of RNA: messenger RNA, we know as mRNA, ribosomal RNA, which we know as rRNA, and transfer RNA, which we know as tRNA. Okay? We'll start out with messenger RNA. These are the instructions to build the actual proteins in terms of the order of amino acids. And this is largely because we don't want the DNA to leave the nucleus. So we transcribe a copy of mRNA off the DNA sequence. And then we go ahead and edit that initial transcript by keeping that's right, exons expressed, very good, and cutting out the introns. The messenger RNA then carries that set of instructions in its final form after it's been edited out through the nuclear pores to the ribosome where it's going to take place doing translation. So, when we get to the ribosome has two major subunits, a large and a small subunit. And the large unit and small subunit have ribosomal RNA in them that help give them the structure that helps to attach to the mRNA and tRNA to allow for the process of translation. Now, we wouldn't be able to actually assemble the proteins because we couldn't make the polypeptide bonds if it wasn't for this last type of RNA, transfer RNA. So tRNA is what actually brings the amino acids in, and its anticodon, that triplet for tRNA, matches up to the codon, the triplet in mRNA, which gives us a very specific amino acid. And we can figure out what amino acid that is by using our codon wheel. But remember that codons are in mRNA, not in tRNA. So in transcription, we're going to take place inside the nucleus because we want to go ahead and protect the DNA. These are the original blueprints. They don't want to go out of the nucleus. They have that extra layer of protection from the nuclear membrane. But we go ahead and make a copy. We do that using an important enzyme known as RNA polymerase, which uses the template strand of DNA to create its RNA counterpart. So RNA polymerase starts copying at a promoter sequence and goes ahead and copies straight through until it reaches a stop. In RNA, thymine is replaced by uracil, Remember T in DNA, but U in RNA. And there are a total of 4 bases that make up our codons, A, U, C, and G. Modified Chargaff rule tells us that A binds to U and C binds to G. Those make up triplets that are what we call a codon. So remember, in DNA, a triplet is called a triplet. In mRNA, a triplet is called a codon. And then later we'll talk about a triplet in tRNA being called an anti or opposite of the codon. So if we look at transcription, remember that transcription takes place inside the nucleus. In the nucleus, the first step we have to do is unwind and unzip. When we unzip, we are breaking the hydrogen bonds that hold the nucleotide bases together. And then we go ahead and make a copy of the DNA molecule using RNA polymerase. Now, RNA polymerase is going to go ahead and copy a complementary sequence. And when it does so, it replaces thymine with uracil because uracil is found in RNA versus the thymine, which is in DNA. Once it has made that copy, this is just the initial transcript, which then needs to be edited. We go ahead and keep the exons. Remember, exon in this case is expressed, not out. And the introns are removed. These are the intervening sequences to produce the final mRNA transcript, which is then shipped outside of the nucleus through the nuclear pores. Let's do it one more time. So again, we have transcription. And in transcription, we're going to go ahead and unwind and unzip the DNA. When we go ahead and do this, we're going to make a new copy using the enzyme RNA polymerase, which is going to put down bases to match complementary to those in the DNA. But as it does so, we still use a modified Chargaff's rule, A to U, C to G. But as we do so, we have to remember that there are no T's in RNA. U, or uracil, replaces the thymine. Once we have done our final edit of that initial mRNA transcript and removed the introns, leaving our exons behind, we then ship that to the ribosome where we're going to do the process known as translation. So translation does take place at the ribosome. And during this process, we're going to go ahead and translate the language of nucleotides into the language of proteins, which is amino acids. So during translation, the cell uses the information from mRNA to produce our proteins. This is because we use the tRNA molecule, which has 3 bases, again a triplet, called the anticodon, which is complementary to the triplet in mRNA, the codon. So the codon matches to the anticodon, and when the tRNA does this, it brings in the amino acid to the ribosome, which is then going to be attached in a specific order according to the instructions on mRNA. The bond in which we go ahead and attach these amino acids together is called a peptide bond. Thus, the finished product is called a polypeptide. This is another example of a polymer and monomer relationship. So let's go over translation one more time. So in translation, we have a special tRNA molecule, which has an anticodon at the bottom and amino acids at the top. The amino acid is very specific to the codon and anticodon pair that is lined up together at the ribosome. The ribosome is made up of 2 subunits, a large ribosomal subunit and a small ribosomal subunit. Those are assisted in their structure by rRNA, or ribosomal RNA. The transfer RNA is going to match up beginning at a start sequence, which is AUG in our mRNA codon. That is going to produce the specific amino acid methionine, which is going to be the first amino acid in the protein. The methionine then will be bound to a second amino acid based on the second mRNA triplet, which matches with the tRNA anticodon triplet. And that bond is going to be what we call a peptide bond. Once that occurs, everything slides down to the left. We kick off the tRNA but keep the amino acid. and bring another tRNA in to bring in another amino acid. When this happens, we keep ratcheting things down, adding more and more of our amino acids until we end up with a long sequence of amino acids kept together by peptide bonds. This is why it's called a polypeptide. And this is not actually the protein, but this is the first stage or first layer of structure in the protein. This will then fold into our secondary and tertiary structures for all proteins, and for our more complex proteins into quaternary structure. It will keep going ahead and adding amino acids until it reaches a stop codon. But unlike the start codon, which does code for an amino acid, methionine, the stop codon does not code for an amino acid and terminates the sequence. Once we reach the stop codon, the two parts of the ribosome separate, release the mRNA, which then becomes recycled, and the polypeptide chain begins the process of folding into the final protein. So here we go. Let's go from the top, from all of transcription and translation together. In the beginning, we start with the DNA double helix. Step one is to go ahead and unwind and unzip it. When we say unzip, we mean to break the hydrogen bonds between the nucleotide bases. Once we've done that, we are going to copy the template strand of DNA. And as we do so, we're going to use the enzyme RNA polymerase to copy through, remembering that RNA does not have thymine. Instead, we replace it with uracil. But otherwise, we go ahead and make pairs, base pairs, according to the modified version of Chargaff's rule. Once that has been completed, we have our initial mRNA transcript. That initial mRNA transcript then is edited to keep the exons, X for expressed, and we remove the introns, inter for intervening sequences. Once that is complete, the final mRNA transcript then goes ahead and leaves through the nuclear pore and heads to the ribosome where we do translation. So transcription takes place in the nucleus, the final product of which is a final mRNA transcript, which leaves the nucleus through the nuclear pore and heads to the ribosome. At the ribosome, we're going to go ahead and match up the mRNA triplet known as a codon with the tRNA triplet known as the anticodon to give you a very specific amino acid. It does this beginning with a start codon, AUG, which codes for methionine, and then it starts forming polypeptides by attaching a peptide bond from one amino acid to the next. These peptide bonds hold the amino acids together while the tRNA is released to allow for another tRNA to come in with a different amino acid and continue the chain. It continues in this fashion until it reaches a stop codon, which, unlike the start codon that does code for a particular amino acid, the stop codon does not code for amino acid and terminates the chain. When this occurs, we now have a series of amino acids connected together by peptide bonds, which creates a polypeptide. That polypeptide then will begin to fold into the different structural components of a protein. The primary structure is going to be the actual sequence of amino acids itself, but then we begin to fold into alpha helices and beta sheets, which creates our secondary structure. How those fit together creates our tertiary structure, which is our subunit. And for smaller proteins, that's all there is. But in more complicated proteins like hemoglobin, we will have multiple subunits that will fit together to produce the final protein. Now I have a quick little video for you to go ahead and watch that will kind of overview some of this and kind of relate it to the genetics that we will do at the beginning of next term. So go ahead and watch this video, and then we'll do a quick little review at the end.
At a molecular level, the two DNA backbones are made of phosphate and sugar molecules. And the substances which join them are rungs of adenine, thymine, cytosine, and guanine. We call these chemicals bases. Adenine only joins to thymine, and cytosine only joins to guanine. The order in which bases occur along a section of DNA defines the types of proteins made in a cell. Think of it as a genetic recipe or code. But proteins are actually made up of smaller chemical units called amino acids. There are 23 amino acids in total, and each one is defined by a group of 3 bases. When joined together, the number and order of these 23 amino acids can combine to make many millions of possible proteins. But with all these letters flying around, it's vital to clearly mark where a gene begins and ends. Whenever sequences like TAA, TAG, or TGA appear, the beginning or end of a gene is marked. These are start-stop signals and play a vital role in genetic engineering. All the genes in a cell of an organism are called the genome, and this is the genome for the tiny bacterial virus X174. It's made up of 5,000 bases, or enough letters to fill a small section of a daily newspaper. In one human cell, the base sequence is around 500,000 times greater. You'd need tens of thousands of newspaper pages to list the entire human genome. Enough to cover many suburbs. The amazing thing is, all this information is held in just one of our cells. In a monumental task, the human genome is now being mapped to uncover the role of all our 100,000 odd genes. This worldwide initiative started in earnest in 1990 and is costing $200 million each year. With so many genes to discover, the project is due for completion early in the new millennium.
The Human Genome Project will characterise all our genetic information. There will be tremendous benefits from this in terms of the understanding of the genetic basis of disease and the development of new drugs to treat such disease.
Okay, so this video is a little outdated at this point. We have since completed the primary objectives of the Human Genome Project, and instead of 100,000 different genes, they identified in humans only about 30,000 genes, a little over that. So their estimates were off in terms of how many they thought we had. So when we start going ahead and looking at this, however, it has some good information reminding us about the relationship of 3 bases represents 1 amino acid, and the fact that we have these start-stop signals. Now, I will also highlight here that at the time, you'll notice that they highlighted all stop signals. So our understanding has improved with respect to this activation or this turning on and off of genes as well. And so this comes from the 90s, and you can hear just how much this cost. It was an enormous undertaking and incredibly expensive, but it has already yielded some good information with respect to certain types of diseases and the ability to go ahead and generate drugs or vaccines to help to combat some of these illnesses or treatments. Okay. So we'll touch a little bit more on this, especially the Human Genome Project and some of these other things when we go ahead and get to DNA and genetics next term. Okay. So when we go ahead and take a look at this, today we're really focusing on practicing with respect to going ahead and making our proteins. So I wanted to give you one more sample, and we'll go ahead and have you go ahead and do this to practice. You can bring it to me to go ahead and have me check it. So here's a very short DNA sequence. Okay, and I want you to be able to use those triplets to convert it into mRNA, tRNA, and then ultimately amino acids. Now, when you're doing this, remember you're going to need your codon wheel to get your amino acids. And remember what that term codon means. Okay, what is a codon? And what does that mean you're going to use to get your amino acid sequence? Okay, so that's what I've got for you today, guys. I hope you have a great rest of your day, and we'll go ahead and talk to you soon. Take care. Bye-bye.